Inside the nucleus lies the blueprint that dictates everything a cell will do and all of the products that cell will make. The information for this is stored within DNA. The nucleus sends commands to the cell via molecular messengers that translate the information from the DNA. Each cell in your body, with the exception of germ cells, contains the complete set of your DNA. When a cell divides, the DNA must be duplicated so that each new cell receives a full complement of DNA. The organization of the nucleus and its DNA. Like most other cellular organelles, the nucleus is surrounded by a membrane called the nuclear envelope. This membranous covering consists of two adjacent lipid bilayers with a thin fluid space between them. Spanning these two bilayers are nuclear pores. Nuclear pores are tiny passageways for the passage of proteins, RNA, and solutes between the nucleus and the cytoplasm. Proteins, called pore complexes that line the nuclear pores, regulate the passage of materials into and out of the nucleus. Inside the nuclear envelope is a gel-like nucleoplasm. The nucleoplasm has solutes that include the building blocks called nucleic acids. There also can be a dark staining mass often visible under a light microscope called the nucleolus. The nucleolus is a region of the nucleus that is responsible for the manufacturing of ribosomal RNA. Ribosomal RNA is necessary for the construction of ribosomes. Once synthesized, newly made ribosomal units exit the cell's nucleus through the nuclear pores. The genetic instructions that are used to build and maintain an organism are arranged in an orderly manner within strands of DNA. Within the nucleus are threads of chromatin composed of DNA and associated proteins. Along the chromatin threads, the DNA is wrapped around a set of histone proteins. A nucleosome is a single wrapped DNA histone complex. Multiple nucleosomes, along with the entire molecule of DNA, appear like a beaded necklace, in which the string is the DNA and the beads are the associated histones. When a cell is in the process of cellular division, the chromatin condenses into structures called chromosomes. The function of these chromosomes is so that the DNA can be safely transported to daughter cells. The chromosome is composed of DNA and proteins. It is the condensed form of chromatin. It is estimated that humans have almost 22,000 genes distributed on 46 chromosomes. DNA replication. In order for organisms to grow, develop, and maintain health, cells must reproduce themselves by dividing to produce two new daughter cells. Each of these daughter cells has the full complement of DNA that was found in the original cell. Billions of new cells are produced in an adult every day. Only very few types of cells in the body do not divide. These include nerve cells, skeletal muscle fibers, and cardiac muscle cells. The division time of different cell types varies greatly. Epithelial cells of the skin and gastrointestinal lining divide very frequently to replace those that are constantly being rubbed off by friction. A DNA molecule is made up of two strands that are complementary to one another. These complement one another in the sense that the molecules that compose these strands fit together and bind to one another, creating a double-stranded molecule that forms a double helix. This double helix can be constructed easily because the two strands are anti-parallel, meaning the two strands run in opposite directions. Each side rail of the DNA ladder is composed of alternating sugar and phosphate groups. The two sides of the ladder are not identical, but are complementary. These two backbones are bonded to each other across pairs of protruding bases. Each bonded pair forms one rung of the ladder. The four DNA bases are adenine, thymine, cytosine, and guanine. Because of their shape and charge, the two bases that compose a pair always bond to one another. Adenine always binds with thymine, and cytosine always binds with guanine. The particular sequence of bases along the DNA molecule determines the genetic code. Therefore, if two complementary strands of DNA were pulled apart, you could infer the order of the bases in one strand from the bases in the other complementary strand. For example, if one strand has a region with the sequence AGTGCCT, then the sequence of the complementary strand would be TCACGGA.